evening, everyone. It's really great to be back here in Edinburgh again and to tell you a little bit about the progress that SkinBioTherapeutics has been making since we were last here in February. We're kind of hoping we get home tonight because when we were here in February, we got snowed in. But nevertheless, it's a great place to get to have an enforced holiday, even if it is an enforced holiday. Okay, so what do we do? So we make new therapeutics for skin, so healthy skin as well as diseased skin. And our technology is based on the human microbiome. And if you're not sure what that is, fear not, I'll tell you in a minute. But, okay, our disclaimer. So just to begin with, a bit of a helicopter view of SkinBioTherapeutics. So we're in a new aspect of microbiome research, which is a good place to be because we're kind of early adopters of microbiome-based technology for skin. We have proof of principle in 3 potential applications that we're developing as a company, but there are many more potential applications. All our technology is supported by our own IP. So we have 3 patent families, and this is all supported by peer-reviewed, published data. And that's because of my background as an academic scientist. So the technology has actually come out of my academic laboratory at the University of Manchester. So the fact that this technology is robust is extremely dear to my heart. Our board is a healthy mix of science and people with commercial experience. So, for example, our chairman's got a track record in delivering growth in private and public companies. Yeah, he's been through exits, he's taken companies through IPOs, etc. Our business strategy is that we're scalable, it's low cost, and we're looking to out-license or partner very, very early on. And we have value inflection points in the near, medium, and longer term. And by near term, I mean very near term. So our first big milestone is a human study, which is actually happening in Edinburgh this month. So very near to a big milestone. So just a snapshot. We came to AIM just about 18 months ago. We raised 4.5 million at a price of 9p. Our current share price is about 21p, so we've done pretty well on AIM over the last 18 months. Market cap of about 25 million, and we've still got lots of cash in the bank. And our CFO is here with us, so he'll be pleased to answer any questions you might have about that. Those are our major shareholders. Some of you may know Probiotics. We technically are a spin-out from Probiotics. With Seneca Partners, who cornerstoned our IPO, and then the University of Manchester, which, as I explained, is where the technology came from, and the 2 academic founders, which is myself and my academic colleague, Andrew Matt-Bain. Okay, so now for the science. So the human microbiome. So I think we all are aware from the great job that the food industry have done for us that we don't just live in isolation as humans. Actually, what we are is incubators for a whole bunch of bacteria. And the bacteria that live in and on our bodies, there are at least as many of them as there are of us. So a way to look at this is that we might only be about 50% human if you think about it like that. I find that quite fascinating. And really, the microbiome is just the name given to all the genes in all those different bacteria. That's all the microbiome is. And the microbiome has got about 1 million genes in it, whereas as humans, we've got about 23,000. So quite a puny number of genes compared to the bacteria that live in and on us. But what that really means is that the microbiome has got a huge potential to be able to change how our body functions. And it does, in fact. Bacteria are fabulous little factories of molecules that can change the way our bodies work. So, probably the best understood aspect of the microbiome is probiotics. And I'm guessing that most people here are very familiar with probiotics. They're in yogurts, health drinks. They've been a massive commercial success. And of course, these are bacteria that usually live in the gut. And they have proven effects in a number of gut-related conditions. So, for example, if you're going travelling and you might get a bit of deli belly, if you take some probiotics before you go, you're usually okay. So they work generally through a number of mechanisms. So they can stop nasty pathogenic bacteria from setting up house in the gut. They're also very good at strengthening the gut as a barrier. So as well as absorbing food, your gut does stop nasty things from getting across it that you might have inadvertently taken in with a meal. And the other thing that probiotics are good at is healing tiny wounds in the gut, such as the sorts you might inflict upon yourself if you drink a lot of alcohol and have a very hot curry. So that kind of makes tiny wounds in your gut, and your gut bacteria or probiotics are very good at fixing those up. So back in Manchester a few years ago, we were thinking about probiotics and the things that they can do. And we thought, yeah, okay, so preventing pathogens, strengthening the barrier, healing wounds, these are really applicable to skin because skin gets infections. Skin is the mother of all barriers. It's the major interface between all your insides and everything out there. It's got to protect us from pathogens, pollutants, UV from the sun, etc. And of course, skin gets wounded. So we thought, if we can find a bacterium that can do those sorts of things for skin, that will be extremely useful in a number of skin conditions. And so that's what we did. So we screened extracts of probiotic bacteria for efficacy in skin. But we had a couple of things that we really wanted from this bacterium. So we didn't want to use these as live bacteria. We wanted to use them as an extract or a lysate, as we call it. And that's because of a couple of things. One is formulation. So I don't know how you keep live bacteria alive in creams and gels and the sorts of things you need for skincare. So we thought if it would work as a dead bacterium, that gets over that very practical problem. But the other thing is safety. So even very safe bacteria that people have been ingesting in their yogurts and things for years, if you put them on compromised skin, they might not be quite so safe. But if it's a dead extract, that's probably very safe. So those were our criteria, that they needed to work as a dead extract. And so this idea of using a lysate or an extract of a probiotic bacterium, this is the basis of what we call our SkinBioTherapeutics platform, which contains no live bacteria. Now, if you've got something that can boost the barrier, heal skin, and prevent infection, it's got a number of applications, and these are the ones that as a company we're targeting. So cosmetics, for example. Many of you will be aware the cosmetics industry is huge and global. There's a cream for everything. If you have got a technology that can really boost the skin barrier, that's a great cosmetic. That's a moisturizer, adds hydration to the skin, and so the look and feel of it will be a lot better. Eczema. I'm guessing most people here will know somebody who's got eczema because it's currently about 20% of at least the pediatric population in the Western world. It's a huge problem, and there's still a significant unmet clinical need for better therapies for eczema than the ones that are currently available. So in eczema, there's a couple of things. The skin barrier is weak, and it also gets invaded a lot by a nasty pathogen called Staphylococcus aureus. So if you've got a technology that can build a barrier, make it stronger, and inhibit Staphylococcus aureus, you've probably got a pretty good, innovative new therapy for eczema. And the other final thing we're looking at is just infection, because, as I said, if you can prevent infection with your technology, that's got a lot of applications. So, a little bit of science. So this is how we do our screening. So the top panel there, that's actually human skin, but it's sitting in a dish in the laboratory. So we get that skin from the leftover bits from plastic surgery procedures. So, yeah. So people that go for facelifts and tummy tucks and breast reductions, that kind of thing, they donate to us the leftover bits that are sort of cut off and would otherwise go in the clinical waste. And we've developed ways of keeping that skin alive in the laboratory. We can keep it going for about 2 or 3 weeks. And it's a great model because it is human skin. It's just not attached anymore. So this is an example of an experiment that we've done. The green staining, this is where we've sectioned that skin and we've stained it with a dye that is a marker for a very important barrier protein in the skin. So just to orient you, this is where I wish I was a little bit taller. The top surface here, that's the surface that you're used to looking at. So you're looking at a cross-section through the skin. Okay? So I hope you can see that in the untreated, the green is not as bright as in the skin on the right that's been treated with the technology. And the reason that's brighter is because that's an indicator that there's more of that important barrier protein in the skin. So what the technology has done is it's put more of this important barrier protein in that piece of skin. And we've gone on to make a cream now with the technology in it, and we can show exactly the same thing with the cream. It's also, we see this effect at doses that are well below the maximum that we can use for safety purposes. So this is a great effect. So to take that into a cosmetic, these are the sorts of things that we've had to do. And this one slide, this is about 18 months of our life now, the people in the company. It's been a lot of work, and just to see it as one slide is, well, anyway, let's just say I'm really tired. But we've managed to achieve all this in 18 months. So the first thing we've had to do is work up a manufacturing and a scale-up method, because it's no point having a great technology if you can't make it at scale. We've done that. We have the technology successfully manufactured now by an external partner. The material that they're producing is consistent to the material that we produce in-house. We've had to take that into some safety studies. So there are tests that you have to do called cytotoxicity testing. And this is the requirement that you have to fulfill before you're allowed to go into a human study. And we've done that. We passed all those safety tests. The other thing that we've had to do, of course, is formulate the technology, get it into a cream, because we need something that we can rub into our volunteers for our study. We've done that. We've got a cream that shows effectiveness, and the effectiveness lasts over a period of time that we need for our study. And we've met all the regulatory requirements that we've had to fulfill in order to be able to do a cosmetic study. So this study is being run this month by Alba Sciences here in Edinburgh. They're recruiting 120 volunteers for us. The volunteers are going to be treated with the cream for 4 weeks, and we're going to take a variety of measurements that are well-known, bespoke, validated measurements of skin barrier function. So something called TEWL, that stands for transepidermal water loss, just basically is a measure of how much water your skin is losing. So obviously, the better a barrier it is, the lower that value is. You lose less water through it if it's a good barrier. Corneometry is a measure of how hydrated the skin is. Obviously, if you build a better barrier and stop water leaving, the skin is much more hydrated. Erythema is just the fancy name for how red is the skin. So redness is important because sometimes if the skin is dehydrated, it gets a bit red and sensitive. So if TEWL and corneometry are working really well, erythema should go down. It should look less red and irritated. Elasticity, it's just basically if I pinch my skin like that and let it go, it should snap back into place straight away. So the better your skin is, the healthier it is, the more it will snap back into place. And then, of course, we're looking at the microbiome, so making sure that the technology is safe for the microbiome. Everybody wants to see that on labels of skincare products these days because people are very aware of the importance of a healthy microbiome. So that's what's going to be happening over the next few weeks right here in Edinburgh. Okay, so moving on to our eczema and also our infection program. So this is the data that we have here. So again, this is the skin. And in this slide, the skin is stained blue, and the red staining that you can see, this is this nasty pathogen, Staphylococcus aureus, which is important for eczema sufferers, but also generally important because it's probably the most prevalent human pathogen, actually, but it's definitely the most prevalent pathogen of the skin. So on the left here, without the technology, you can see that there's red staining right the way through because the pathogens basically invaded the skin and gone all the way through the different layers. On the right, you can see that there's hardly any red staining at all, and that's with the technology. And so this technology is acting as a barrier to stop that pathogen from being able to invade the skin. So we like that data a lot. It's a nice visual way of showing how great this technology is at inhibiting pathogens. This graph, this is just looking at how the technology stops pathogens from growing on the skin. So the blue line is what happens if you put Staphylococcus aureus on the skin and just let it go. And you can see that the line goes up, and that's showing that the pathogen is growing really well. But the red line, in the presence of the technology, that red line is flat, at least for 5 to 6 hours. So it's basically stopping the growth of this pathogen. So what this data also tells us is what our dosing and frequency of application would be. So we'd be looking at a 3 times a day application. And this data is applicable to our eczema and our infection programs. And currently, we're defining the regulatory pathway for use of our technology in this way. Okay, so the main highlights since we IPO'd, and if you remember, we IPO'd just 18 months ago, so we've got production now of cosmetic-grade SkinBioTherapeutics at scale by a third party. The second one, freeze-drying method established. This is really important. So the freeze-dried version of the technology, this, when we produce it in the lab, it's a liquid, and that's not really very good because you can't transport liquids at volume and they're a bit harder to work with. But being able to dry it and create it as a powder, that gives it shelf life and it's ease of transport. And it's that version, that's what we're trying to commercialize now, that freeze-dried version. So we've cracked that and we've shown that that works very well. It works just as well as the stuff that we make in the lab. We've shown safety, we've got a stable formulation, we've made a lot of scientific progress, including getting patents to grant in Australia, Russia and New Zealand. We've got 17 patent applications out there at the moment, and the fact that we've got 3 to grant, that gives us a good idea that the others are probably going to go to grant as well, because most patent offices work off the same information. We've also had a grant awarded in conjunction with the University of Manchester, and this is to start doing some studies that will help to generate a pipeline for us, so new technologies potentially coming into the company. And commercially, we've had a material transfer agreement with a large global consumer goods company. So the deal there was we signed this agreement, and that gives them access to our technology, not exclusive access, they just get some access to it, to have a look at it in some of their potential applications. So that was great for us because we're a tiny company, they're a major global force, so it's great that our technology is being tested in-house. So this is what our pipeline is looking like. For the cosmetic, obviously we've got proof of mechanism, we've done our formulation, we're into our proof of concept studies this month, and once we come out the back of that study, we can seriously start to commercialize this technology. The value of having data in humans cannot be overstated. That is absolute dynamite to have data in humans. So we always knew that we weren't going to aggressively commercialize this technology till we got our human data, because that changes the face of the conversation that you can have with potential commercial partners massively. Our other 2 programs, because the regulatory pathways are a little bit more complicated, they come on board slightly behind because we have technical files, etc., that we have to produce before we're allowed to go into human studies for those 2 applications. So our business approach, obviously we've got a platform-derived technology that's got lots of applications, and our aim is to continue to build a portfolio of IP and also supporting science, and that's done back through my academic lab at the university, because that's the best model for us. We're not in the business of building huge laboratories ourselves. We don't need to. We know that the scientific rigor behind our technology is driving partner engagement. People like that this is a technology that's been based on, that's been published, the data of which have been published in peer-reviewed scientific journals. People really like that. There's lots of licensing potential, and of course, as I've said, our initial market focus will be that cosmetic application once we get through our study. So our outlook really for the next few months, it's going to be data analysis of that cosmetic study, continue our commercialization discussions. We are talking to potential partners, but not making any commitments at the moment until we have that human data. We're going to continue to progress the regulatory pathway for our infection and our eczema programs, and because our company is very much founded in science, we're going to obviously continue the scientific progress of the company. So in summary then, we have a technology that's based on robust science, and we're targeting large global markets in terms of its applications. We have a scientific team that's got close ties to industry, and we're supported by an experienced board with commercial experience. We have a scalable model, which is low cost. We operate virtually, and we're looking to partners to actually commercialize the technology. We're not looking to take anything to market ourselves. That will be done by co-development or by licensing of the technology to commercial entities. And we have near-term value inflection points, and just leave you with the thought that we are a very early mover in applying probiotic extracts to skin. Thank you very much for your attention.
So Super. Same form as before. Any questions, put your hand up and wait for Peter, please. Gentleman here.
Your penultimate, I think it was, RNS, when it was issued to the market, seemed to sort of prompt a sell-off. Was it to do with the freeze-drying side of things?
No, it wasn't to do with the freeze-drying. The freeze-drying is absolutely nailed on, and that's what we're trying to commercialise. I think we released an RNS that was supposed to sound a little bit positive, but in it we said we'd got 24 hours activity in the cream, and I think people took that to mean we had only got 24 hours activity in the cream. But we've subsequently released an update saying that we now have activity that we need for our study, basically. So we're good to go.
Perhaps just the last one then. With your human trials at the moment, what happens if they don't go to plan? Where do you go then?
So this is a cosmetic study. It's not a pharma study. So it's not a binary output. With pharma studies, they either kind of work or they don't. It's very binary. It's not like that for our study. It will be, and we've got a lot of endpoints there. So I'm pretty confident that something will work. And you know what? If it doesn't, there are lots of things that you can do around that in terms of the formulation that you use, etc. So I'm not worried about that. The main worry has been getting us to a human study, but we've done that. So the pots of cream went off in the wagon today to the various test houses. So I'm very happy today.
So in the future, you're not going to have to go through the sort of normal phase 3 trials that pharma companies have to go through? So
for our eczema program, for example, will almost certainly be a medical device type of play. We're not intending to create an RX prescription medicine type of technology here. So for a medical device, obviously the regulatory requirement is higher, but it's not as high as it is for a pharma product where you do have to go through the phase 1, phase 2, phase 3, etc. So no, we're not really intending to do that.
Could you explain the difference between a pharma product and a medical device product?
So for a medical device, the regulatory requirement is that it has a primarily physical mode of action. So we've done an awful lot of science around the mechanism by which the technology inhibits Staphylococcus aureus, and we know it's stopping it from sticking to the skin. So that's primarily a physical mode of action. So that's why we think we can get it through as a medical device. But obviously we've to produce our technical dossier and get the required approval for that.
So could it be then that if you actually did get a license with a major pharma, that they would take it through?
That would be the plan. We're not really intending to do that ourselves. We want to partner with somebody that's geared up for that sort of thing. Yeah.
Any other questions, please? I had a couple. From the study which is due to start, how quickly do you then get the data back and process it, analyse it? When would you expect to be able to report back from that to the market?
Yeah, I mean, I think we're looking realistically. This is actually a big study. We doubled the study size. We had an RNS out where we said we were going to double the study size. So obviously that doubles the length of time needed for data analysis, etc. And we've got a lot of endpoints as well. And also because of the way they recruit, so they don't start everybody off on day one because there's 120 of them. So they recruit them in batches. So I think realistically we're probably looking towards the new year.
Okay. And so the different endpoints, so earlier on in the presentation you talked about things like elasticity, redness of skin. So there's all that range of different things that the product could then, you could say it's great for this one or better for that one.
Exactly. And part of the reason that this kind of speaks to your point as well, part of the reason that we double the study size was because the more people you've got in the study, the more you can start to stratify the data for things like age. So it might be that this technology is only moderate for the general population, but actually it might be fantastic for the over 50s, for example. So with a study size that's 120, that's a bit better, a bit bigger, you can start to look at that. You can start to subdivide the data that's coming out and start to see some trends that you wouldn't see if you were doing the typical size of study, which is more like 30 people for studies like this.
Any other questions? Otherwise I've got one final one. And that's just how much time do you still get to spend in the lab? Are you still in the lab at all, or are you spending your whole time kind of running the business?
Me personally?
You personally?
I barely know the way to my lab anymore. And when I walk in, they all walk out again because I generally cause chaos these days. No, I'm a voyeur of what my team do. So my team are fantastic at the University of Manchester. So they do all the lab work, and I just have a look at the data and get to report to you guys and generally run the business. Yeah.
Lovely. Okay, thank you very much indeed,
Thank
Kath.
you.
Thank you.